All right. So we've talked about mitosis. And the key thing to remember about mitosis is that it's a cell division that produces the same genetic combination. So in a sense, it's like cloning. It's a clone type of reproduction, which means you've got the same genetic material every time. And of course, this works for lots of organisms. They can reproduce this way quite effectively. And I think the example we had talked about was, a strawberry plant, which grows and does have flowers, so it does do sexual reproduction as well, but it can also reproduce in such a way where it starts to grow what's called a runner, which runs out over the ground like this, and everywhere it touches the ground, it will sprout little roots, and a little plant will grow. And because these plants have just grown off of this extension, there's been no exchange of genetic information, so they are genetic clones of each other. This is a form of vegetative reproduction. Many plants can do things like this. But even animals, a planarian worm is a little worm. It looks like a little arrow. It's got a little arrow-shaped head. And if you cut the worm, right, if you slice him in half, like that, what'll happen is the top part will start growing a new tail, and believe it or not, the bottom part will start growing a new little head, and these will grow into two completely new planarian worms. But there's been no exchange of genetic information. It's the same cell. So it's mitotic divisions that cause this kind of reproduction to happen. And it works quite well for many things, but there is an advantage to having an assortment of genes that changes every time. Genetic variability is quite low with this kind of asexual reproduction. Genetic variability is low. I could put that up here. We sometimes call this kind of reproduction asexual reproduction because it doesn't involve the exchange of information and the formation of special sex cells. Now, there is a little bit of variation possible here because even in mitosis, there can be some aberrations or mistakes that happen that do create small amounts of variability over a long period of time. So it's not zero, but it's fairly low, right? It's fairly low. Genetic errors can happen in the mitotic process. Sexual reproduction is a whole different ballgame where we change up the game to allow for an exchange of information, sexual reproduction. And this developed, and when it did develop, it offered more genetic variability, as we'll see. And genetic variability is very important because if you have a species, if all the individuals are sort of genetically the same, then particular stresses in the environment, like diseases and things, will affect them all pretty much the same way. So something that's lethal to one is very likely going to be lethal to all of them, right? So this is the situation with the asexual reproduction. But if you have a species where there's some genetic variability built in, then some of the combinations of genes that you get will result in slightly different individuals. And these different individuals might have the ability to withstand a particular stress in the environment more easily. So when the stress occurs, some might end up dying, but the others will have a slightly different genetic combination which offers them a slight advantage, meaning they can survive. So more genetic variability can translate into a better chance of survival for an entire species. So it does tend to be a very popular way of reproducing in the sort of latter part of the evolutionary story. Now, sexual reproduction is old. It originates quite a ways back. but of course not quite as far back as the asexual reproduction that we see. All right, so how does sexual reproduction work? Well, it works with the process of meiosis. Because the trick is, if you're going to exchange genetic information, you're going to combine two cells to do it. You have to have a way to cut your chromosomes in half. Otherwise, they'll just keep doubling, and you'll end up with double, 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 and before long, hundreds and hundreds of chromosomes, which there's no room for, and also makes for too many instructions and nothing that can survive. And I should point out that there is a way to exchange genetic information that bacteria use. Essentially what bacteria do is they come very close together, and their membranes can sort of fuse or open up a channel, and bits of DNA can be moved from one to the other. This is called conjugation. So it is a form of exchange of genetic information. And then each of these would go their separate ways, and then they would reproduce with the new combination by mitosis. So it's still a mitosis-based kind of reproduction. So it's not sexual reproduction. In order to be sexual reproduction, you have to produce a specialized sex cell called a gamete. So although this is an interesting form of exchanging genetic information, meiosis does not apply to that kind of exchange. It's just basically getting together and trading DNA. Whereas in meiosis, we're going to make specialized sex cells that are designed to recombine with each other from different individuals. Okay? So let's look at meiosis in detail. In order to understand meiosis, you have to understand that the idea of the homologous chromosome. So I'm going to draw this cell right here. And I'm not going to draw the nuclear membranes, because as the cells divide, the nuclear membranes disintegrate, right? And they go through the processes. So we're going to try to put some chromosomes in here. A red one, a green one, a blue one, a purple one. Let's see if we can get two more. Maybe this orangey colored one, and maybe this yellow one. So we got 6 chromosomes. Of course, most cells have far more than 6, but for our drawing, we can illustrate the process with 6, and we can understand that it would work the same way with 46 or 27 or whatever number they had. Okay. So this is the full complement of chromosomes, and there's a name for this. So the number of chromosomes here is kind of important to keep track of. The number of chromosomes in the mother cell is 6. We call that the diploid number. That's an important word, the diploid number. The word di reminds us that this particular genetic count includes 2 homologous pairs, or 2 homologous chromosomes, right? So let's talk about that to remind us about what homologs mean. In this cell, I'm going to draw some comparisons. The red chromosome and the purple chromosome are going to be homologs. Sorry. The blue and the green are going to be homologous or homologs, and the yellow and the brown are going to be homologous. So what does that mean? Well, what it means is that while the 2 chromosomes are slightly different, their instructions are for the same particular thing or the same job. So we use the example where, we have a society, and these 2 chromosomes are firemen. These 2 chromosomes are construction workers, and these 2 chromosomes are dentists or something. Now, they might be slightly different. One of these firefighters could be a girl, one's a boy. They have a slightly different, approach to how they fight fires, but they get the job done. Right? A biological example could be where the red chromosome, in this case, has genes that tell it to produce a chemical called melanin, a protein, which makes skin and hair and eyes turn darker. And so does this one. It would also have, in the exact same spot, too, a melanin instruction gene. but they could be slightly different. One could code for a heavier production of melanin or more melanin, while the other produces less. Or, in fact, it could be none or all. And so they're slightly different in how they go about making this melanin, but they're homologs because they both have that job. They both make melanin. And that difference is going to become important when we get into the genetics. Okay? So what we have to do is, if we were going to separate these chromosomes, we'd have to make sure, if we separated them, that we got one of each job in each group. So when we make two groups here, if we want to separate them, we have to have a red one here, and we have to have a purple one here, because if the red and purple are together, that means there's two firemen in one cell and no firemen in the other. There's going to be two melanin-producing genes in one cell and none in the other. So we need to have them separated. So that's what the idea of homologs means. So when you separate, the first step is to separate the homologs to make sure that you're not leaving out any jobs in either of the two cells. And to do that, we have a division called the Meiosis One, the first division. So let's do Meiosis One together. Let's go back to black. So we call this division Meiosis One with a Roman numeral 1. And essentially what happens is we start with the phases. The phases, we use the same names as we do for mitosis, but we use the Roman numeral. So this first phase is called Prophase One. And in Prophase One, we have a cell. And what happens is the nuclear membrane begins to disintegrate, just as before. And of course, you'll see how the chromosomes I drew up here were single, just to show you the numbers. But we have to remember that when the cell's getting ready to divide, each one of those chromosomes copies itself. So they're like a double wiggle, right? So we have to draw them as copies of themselves. So what we would have in here is a red one that also has a copy, and a blue one that also has a copy, a green one with a copy, a purple one with a copy, a yellow one with its copy, and the brownie one with its copy. So it would actually look like that because they've doubled themselves up, made their copies, right? That happens in the S phase before all of this begins. So in Prophase One, we see the disintegration of the nuclear membrane. We see the centrioles. I'm running out of colors for structures here, so I'll just use black. The centrioles, right, start moving to the poles of the cell, and they're going to be where the spindle fibers attach. But what happens here in the next one, which is Metaphase One, is that the chromosomes are lined up with their homologous partner. And this is incredibly important. The homologous partners must be together in a sort of a group. So what that means is, in our picture of our cell, and I'll go back in a minute if I have to, we said the red guy was homologous to, let's go back up here, the purple one. See, the red and the purple are the ones that do that particular job. So the red and the purple have to line up together, and they actually stick together like that in something called a chiasma, which is a joining of the two chromosomes sort of temporarily together. Now, there's really two chromosomes here, but because each one has copied itself, there are four little chromosome tails, right? So there are four little chromosomes or chromatids all attached in this, and so we call this a tetrad. It's called a tetrad, T-E-T-R-A-D, tetrad, when they link up like this. Then we have to put together, I think the yellow chromosome was homologous to the brownie-colored chromosome. So we'll put the brown one together with the yellow one. And then we had the blue and the green that were homologs, joined together like this. Okay, and they're lining up sort of in the middle of the cell, and of course the centrioles are in place, and then what happens is we see the spindle fibers, almost fully formed like this. And you can kind of guess what's going to happen. Those spindle fibers are going to pull on these, and they're going to pull one of each job, one of each homologous pair to either side of the cell, making sure that all of the jobs are represented in the two daughter cells that would be produced. So then we go to anaphase. All right, we just bounced around here. Somebody wanted the spelling of the chiasma, which is the sort of the joining spot there. So we put that on the board and caught up with our pictures. Okay, so now let's go on to Anaphase One and see what happens. In Anaphase One, the cell begins to elongate. So we get a picture of the cell sort of elongating like this. And then I'm going to have to look at what's on the bottom here. Purple, brown, and green. Okay. So the purple, brown, and green homologs are being pulled downwards. So we would see the purple one. Whoops. Come on. Purple, brown, and green. Purple. And it would be being sort of pulled down like that. And then the brown one, I think, was next. So we'll put the brown one in, and it would be pulled down. Go away. Go away. Can't get it to disappear here. Why is that? Okay, I'll just move this underneath it. So the brown one will be pulling this way. And then the other one was what? The green one, right? I'll just double check that I've got them on the right side. Yeah. And then going to the top is the red, the yellow, and the blue one. So the red is being yanked up this way. And the yellow, similarly, and the blue yanked up this way. And remember, there are still two copies. We have not separated the copies. All we separated are the homologous pairs, the ones that do the same job. That's the whole point of the Meiosis One division. So these are being pulled by the spindle fibers towards the ends of the pole. And then we have, of course, Telophase One. So if I jump down to that one, Telophase One is the situation where the cell now is nearly finished with its separating. And so we have a red, yellow, and blue in the top. Red, whoops, that's not red. A red chromosome that was pulled this way, a yellow chromosome, and a blue chromosome. And that leaves us with, on the bottom, I believe, a purple chromosome, and a brown chromosome, and the green. So then eventually what happens is there's cytokinesis here, the pinching in of the membrane, and we get two cells. So at the end of telophase, two cells. And they have half of the original chromosomes that the mother cell had. There's only 3 in each, whereas the mother had 6. But it's a very select 3. It's 3, but it's the 3 that do all the same jobs as the other 3. So the homologs are separated. Okay? And that's the end. Now, normally in mitosis, that would be the end of the story. But these two cells are now going to do a second division. Now they're going to divide again, and this time the copies are going to be pulled apart. So we'll do what we call Meiosis Two, which starts with these two daughter cells over here on this side. They move on to Meiosis Two, the second division. I'm only going to show you one division, right? Well, why not? We could do both. So let's put our two cells here. We would have Prophase Two is the starting point where we have the two cells from before, and we have the red, the yellow, and the blue, like this, and the purple, the green, and the sort of brown color, right there. So that's the end of Telophase One is essentially the start of Prophase Two. But of course, we move to what we would call Metaphase Two on this side, Metaphase Two, and the chromosomes would be lining up in the middle of each cell, getting ready to be pulled apart. Red, yellow, blue. And on the bottom, we would have green, brown, and purple. And of course, the spindle fibers would be forming again. Oops, that's supposed to be black. The spindle fibers are now forming, getting ready to pull apart these copies of the chromosome. So in the second division, it's the chromosome copies that are being separated. In the first division, it was the homologous pairs. Okay? So now what I'm going to do is kind of go in a circular motion. I'm going to draw arrows kind of going around like this. And I'm going to do the next stage under here, which will be Anaphase 2. Anaphase 2. And of course, it looks very much like Anaphase looks. I better open up my page a bit here. The cells would elongate as the spindle fibers pull apart. And so we would get a separating. We would get a green going one way and the bottom one. Whoops, that's upside down, sorry. Oh, that's right, that's good. We would get the brown one being separated and the purple one being separated. And of course, in the upper one, we would have had the, let's put them in the same order, red, yellow, blue. So the red copies are pulled apart, the yellow copies are pulled apart, and the blue copies are now pulled apart. And we can just add in some of our spindle fibers here doing the pulling. Anaphase 2. And then, of course, what happens is each of these will split in telophase. So we can draw little arrows over here. We can call this Telophase 2. And we will get the hourglass shapes as the cytokinesis begins to, or finishes off, really. And what we would see is we would have a cell here with one red here, a yellow and a yellow, and a blue and a blue. On the other side, a green and a green, purple and a purple, and a brown and a brown. Which would then lead us to the final result once they completely pinch off. These particular two, let's go down again. Whoops, let's go up a little. Right, let's swing down around here to the final situation after telophase 2, where we have 4 daughter cells. And the 4 daughter cells on the top have red, yellow, and blue chromosomes. And the bottom has the green, the brown, and the purple. And of course, they would a new nucleus would form. Purple, right? So the nucleus, just like it does, would reform around here. And now we have produced these complete finished daughter cells. So these are called the daughter cells. But remember, with mitosis, you produce 2 daughter cells because it was just one division. Mitosis is almost exactly like this second division. But we get 4 because meiosis adds that first division where we separated the homologous pairs. There was no need to do that in mitosis, but there is a need to do it because what we have here are we have cells that have half the number of chromosomes as the mother cell. That's important because if they're going to recombine by joining, that'll put us back to the normal number. So half the number of chromosomes. And there's a name for that too, chromosomes. We call that the haploid number. So half haploid number. So in this case, the haploid number is 3. Our diploid number was 6. But of course, these cells, what they are sperm and eggs. So we have a name for these cells too, a specialized name. These cells are called gametes. They are different from all the other cells in our body in that they have half the number of chromosomes. They are haploid cells, we call them. And these gametes come in basically two kinds, a sperm cell and an egg cell. So we call them the sperm and the egg. So a sperm has been made by meiosis, and an egg to have half the number of chromosomes. And of course, they're designed to rejoin together. That's the whole point. A mother produces an egg and a father produces a sperm, and what has to happen is the two have to be brought together, to fuse together, so that their 3 and 3 will join into one cell, which will have 6 again. And we're back to where we started with, to have a normal organism, right? That's what they're designed for. Now, I want you to realize that if we go back to our pictures, some important things to understand. All the way back here to Metaphase One. Variation can occur quite easily because if you look at how these have lined up, there's no rule that says that this yellow, or let's do the red and purple chromosome, would line up this way. It's quite conceivable that it would line up the other way, upside down, with the purple one on top and the red one underneath. And if that were to happen, that would change which direction they went. The red would go down, the purple would go up. That would mean at the end here, we would get our gametes. Let's go right to this. The ones with red, instead of the combination of red, yellow, and blue, those reds would be the purple that went up, and the purple down below would be the reds. And so we get a different combination of those 3 chromosomes. The number of combinations, of course, is immense when you start looking at 46 chromosomes in a human and how they could all line up and they could all be flipped in a different way. So the number of eggs and sperm that you make, the different variations that you could get while still having one of each homologue are huge. The other important thing that happens, before we go any further, happens right over here. Let's go back to this Metaphase One. You see these chromosomes, how close they are together. Let's redraw this blue and this green chromosome closer together. And they're joined at the chiasma, sort of like that. Well, what can happen is these tails can twist and turn and get involved with each other. So right here, a piece of this, they might twist, and one might just detach from one and attach to the other, which means you would get a blue chromosome with a little green piece attached to the bottom that it sort of traded with the other chromosome, and you'd have a green one with a little blue piece sort of attached to the bottom. And that means that when they separate, although the green one's going one way and the blue's going another, now we have a little bit of blue going with the green and a little bit of green going with the blue, which creates even more variations in the possible genetic combinations. This is called crossing over. And it's when in the tetrad, it's when the 2 homologous pairs actually trade parts, because they're just molecules, remember? So it's not that difficult for molecules to sort of dislodge from each other and lodge onto other nearby molecules. Crossing over. Again, allows for more variation. So the end result is daughter cells, sperm and eggs, and you could have so many different combinations of genes here, even though you always have one of each homologous pair represented. And that's what produces the great diversity of genetic information in sexual reproduction. And that's the story of meiosis. So we'll stop this here, and then we'll continue on a little bit later in the next video.